Pleasure to be here today in Nebraska. I've been having a very lovely time in the Midwest, which I'm a big fan of in general. I spent 8 years or so in Madison, Wisconsin, three years there, and almost 5 years in Chicago. So it's great. I think you're lucky to be here, away from all the noise, and focus on science and innovation. Are we rolling? Oh, I have to double click, get myself up there. Excuse me a moment. Okay. Great. And I'll just get rid of this. Just click it away. Okay. So this is a penultimate version of the talk. I have two key drives that are identical. It's not a very good idea to have two exact key drives. I think I brought the wrong one here, so this is a next to the last version. The formatting might not be perfect. I've been at so many pharmaceutical companies because originally I was trained as a neuroscientist, behavioral neuropharmacology. I developed an allergy to rats and mice and rodents. My second year of grad school was pretty severe anaphylactic reaction, a delayed response. So I would do behavioral studies, and I'd go home and I would be all congested, and I thought it's the hay fever. It's upstate New York, but it ended up being the animals. So I went into neurochemistry, and then I had a penchant for automation. I was good kind of with computers and juggling data and tuning my own car. Remember the days when some people, we used to tune our own cars? I think not even an engineer could tune their own car today. So I started to become interested in making supporting medicinal chemists more efficient. It would sometimes take a week to get back data. So I got involved in lab automation, and then I started to read the literature. This is what, 1989, 1990, so I'm giving away my age, about this thing called high throughput screening, where you could screen thousands of compounds a day instead of 10 a day and get data back and figure out which one of those were drugs. And everyone bought into this. It was roughly around the time Genomics, high throughput Genomics was coming out, and every company wanted someone to set up a screening lab, so I traveled around the United States and got glimpses of different types of pharmaceutical organizations. GD Searle, where I started, was owned by Monsanto, and that was right around the time the agricultural companies, ChemAgs, had these large chemical libraries that they used to test for herbicides and for pesticides. And they read some of the news stories about high throughput screening, where you could screen thousands of compounds a day and figure out which ones were the drugs. And they said, well, we've got a lot of compounds, so maybe we should get into the pharmaceutical business. So they all bought up pharmaceutical companies. Monsanto bought Searle, and Dow bought Letterly, and Kodak bought Sterling, and so on and so forth. And then after about 5 or 6 years, all the chemical companies realized, oh my goodness, this is much harder than we thought it was to actually get return on investment, to actually not just figure out what the right drugs might be, but to actually successfully develop them. So that's the background of the talk. And I'll show you some statistics about the pharmaceutical industry. I hope you'll walk away from this understanding the difficulty, complexity, and how the odds do not favor success in drug discovery, which is why when someone does succeed, you call that the billion-dollar molecule. There's probably no industry as a whole that has a higher profit margin than the pharmaceutical industry. So that's one take-home message, and that's an overview. The second take-home message is these days pharma is going through some turmoil. It's an industry has been disrupted because of high throughput screening, because of high throughput technologies. Some say a focus was lost on phenotypic pharmacology, good old-fashioned classical pharmacology, understanding mechanism of action. And this is why pharma is having an innovation gap now. Nonetheless, I think I'm here today at the age I am, probably because of pharma. I think at the turn of the century, from the 19th to the 20th, the major cause of death was infections. And that was probably 40% of death. Today the major cause of death is cardiovascular disease. That's a disease of aging. And will we ever be able to fix getting old and live forever? Maybe the answer is yes, but most likely the answer is no. And even cardiovascular disease, ion channel blockers, different types of blood pressure medicine, these have transformed human health. So I don't want anyone to walk away thinking that this is a negative talk on the pharmaceutical industry. It's not. This is just kind of not the best time. You can think back maybe 10 years ago or 20 years ago to Detroit, which is a dinner conversation. The automobile industry went through a tough time as well, and seems to be recovering. So that's the second take-home message. First one is much harder than it looks. The second one is it's worth it. It's worth it. And the third one is I think that as we try to solve diseases that seem to be unsolvable, I don't think pharma will ever do this on its own. They can't be an island unto themselves. Their checkbooks are running out, and their talent is also running out. As they downsize, they lose the classical pharmacologists and the great medicinal chemists. I think they're going to turn to academia. And I think open innovation is something we're hearing as a solution to innovation in pharmaceutical R&D. So those are the three messages. I'll see if I do a decent job communicating it. It may seem like a lot of data and statistics, but it's actually like Time Magazine or The Economist or Wall Street Journal kind of stuff. I think everyone will be able to get it. I hope. So the outline, I'm going to show you, and I'll since I'm a high throughput type of scientist, I'll see if I can give a high throughput talk. I'll try to go through the slides quickly. The evolution of United States academia, which has been called by The Economist as an example, as a golden goose of innovation. Biotech industry, computer industry, wouldn't be here today if not for academia. I'll give you the history of that and how the NIH was formed. What we mean by pharma's innovation gap, I'll explain statistically and operationally what this means. How I try to solve this, and my own humble way with something called double-blinded drug discovery, taking the term. I think it's like a ghost world out there. I don't know what to believe anymore in popular literature. I don't know what to believe in scientific literature, but I do believe in the double-blinded study, clinical trial, prospective. When neither side knows what the outcome is going to be and neither side knows what the treatment is, and you do carefully controlled studies like that, when I see the data from there, I tend to believe it, and I don't believe anything else. So I use double-blinded just at the earliest stage of drug discovery. And I'll explain to you what that model is, an open innovation model. And then a couple, maybe 2 or 3 slides, maybe a dozen on drug synergy. And I think that maybe the next wave of research could potentially be, nutraceuticals, and particularly combining them with drugs to get a therapeutic synergy. I think this is an undermined area. I'm not aware that there's any literature at all on it. And then a summary, which I think I've just given you. So evolution of American universities, I think, came from Enlightened legislation. So I'll give you maybe 1 or 2 historical slides, and then I'll explain to you how the government here has, made some, maybe they were mistakes because they turned out fantastic. It's very rare that legislation turns out great in every way. When it comes to government supporting, sort of applying, translating research in academia, there's some just incredible acts. So the origin of universitas refers to a cooperation of persons associated into one body. And the University of Texas has emblems of the major historical landmarks of universities in one of their campus buildings. The University of Bologna in 1088 defined the freedom to explore. That's what a university is. And the right of a traveling scholar to go anywhere they wanted and should be accepted to different universities in the interest of education. Wouldn't that be if they were great today? You could just say, I'm a little tired of Nebraska, I'd like to maybe go to University of Hawaii, and they go, sure, come on in, and there would be this equilibrium of students through universities. I think that would be great, but probably won't happen. And the freedom to think unfettered from religion, I think is a major concept. So the freedom to explore, that's what we think of as universities being a facilitating, and freedom to think without any kind of hindrance. So interestingly, the settlers came off the Mayflower in 1620, and then within 16 years, I mean, first they came with nothing probably, or very, very, very little. They had to build houses, they had to, settle unsettled land. Yet within like 15 years or so, they established Harvard. That's how important. So religion was very important to the settlers, And education was very important to the settlers. It took after Harvard about 50 years for the next university to come about. And then Yale pretty fast, and then there was another 50-year gap. It'd be interesting to look historically and understand why there were these gaps in between. But then from about 1750 onwards, a steady stream of Ivy League universities came about. So these are the colonial colleges, most of them considered Ivy League colleges today. But there are many more universities than this in the United States, of course. And that's because of Justin S. Morrell and the Morrell Land Grant Act of 1862, where the United States government, I think, gave every state something like 50,000 acres, I forget now what the exact amount, 30,000 acres, and said you can do whatever you want with this land, you could sell it, you can build on it, as long as it goes to education. And what we want is to train United States citizens in the agricultural and mechanical sciences, A&M universities. So that's where they came from. And if you look at the world's top 20 universities, first what's interesting is that 16 of them are in the United States, 3 are in England, that makes 19, and then Sweden has 1. So that's pretty interesting to have such a density of universities. Of the 20, of the 16, only 4 of them, a quarter, are the Ivy Leagues. The rest are actually, or many of the rest are state universities, these A&M universities. And of course, you're all sitting today in one of these A&M universities. And Lincoln University of Nebraska just celebrated, I guess, very recently, I don't know what year this was, I guess 3 or 4 years ago, 150 years of the Morrell Land Grant. Interestingly, Morrell wanted to pass the Land Grant Act under the Ulysses S. Grant administration, but Morrell insisted that the act had to include, non-denominational, and it had to allow African Americans also to go into the universities. And Congress refused to hear of it, so it took Abraham Lincoln to ram this bill through. And during the time of the Civil War, and some think it was a minor factor in upsetting the South even more. We'll jump a little further to 1875, and this is the first president of Johns Hopkins, Daniel Coit Gilman, who, my recollection is also a great physician and wrote a lot about medical science. What are we aiming at? The encouragement of research and the advancement of individual scholars. And this is the interesting point, who by their excellence will advance the sciences they pursue, sure, but also and the society where they dwell. So he was the first individual who articulated the concept that a university ought to give back, that the people who are graduated from university ought to go into their own community and make that community a better place, and that that community should also support the university. So that it wasn't just an Ivy League, gentleman and gentlewoman affair. This was actually applying your knowledge to the entire community, and also implying economic development as well. So this was a radical concept back in 1875. We'll jump to 1945, and this man, Vannevar Bush, a doctor, PhD, Vannevar Bush, not related to any of the Bushes we know today, really championed science as the endless frontier, and that the science should serve society, and in turn, we hear it again, society should provide financial support to assure the advancement of scientists. He wrote a white paper for Franklin Roosevelt, who was president at the time, championing this mightily, and to Congress, and it's due to him. Many people think Department of Defense, the NIH, Department of Energy, EPA, NSF, all of these agencies were articulated in the white paper. And of course, the NIH, most important of all in terms of the amount of money allocated to them, and then they disperse for scientific research, mostly medical research. Of course, the project that validated public-private partnerships can be transformative was the Manhattan Project at that time. So the NIH today is 27 institutes, nothing else like it, I think, anywhere in the world, and the budget as of 2014 is just about $30 billion. Looking at the budget, many of the room may be aware of this, it peaked at around 2002, 2003 in terms of real dollars. So this is the budget. I think under Clinton and partially Bush, but it was under Clinton that Congress agreed to double the NIH budget, that literally from around 14 billion, I think, where it was during the about mid-Clinton administration, and said, okay, over a period of about 10 years, we'll increase that 2x, which was just fantastic. And there was an explosion of scientific research, I think, in the United States, and support for it, and colleges grew and medical centers grew. But these things, what goes up must come down. So then it gradually increased, and now it's remained relatively flat, starting to dip down a bit in terms of real dollars. In terms of constant dollars, constant $95, you can see it's quite a decrease. And when they say constant $95, I think they're talking about general inflation, which, I don't know, on average has been relatively low, especially the last decade. But medical inflation, scientific instruments, reagents, I think you all know, it's probably low double digit, or even today, it's still 5 or 7%. So I think, and if you look at, do I have some text over here? I don't. I think the last version of the slide had some text over here that showed that the amount of R01 grants, the major 5-year research grants, that's the bread and butter of every scientist in every lab, has decreased by almost 40% since about 2003. So these are tough times in academia. And I'll soon show you that tough times in pharmaceutical industry. So I think you can put 2 and 2 together, what a solution might be. Bayh-Dole Act of 1980. Before the Bayh-Dole Act, if you made great discoveries, you couldn't patent them. The government owned all rights to all patents. So guess what? Nobody patented anything. Nobody licensed anything. So there was no tech transfer office before the Bayh-Dole Act in universities. So these 2 guys came up with the brilliant notion, vest scientists in their own discoveries, let them own title to their patents, and let universities own this as well and license it to industry. And that started the biotech industry. That started all the concepts today that seem natural and that have always existed were due to these 2 individuals in the Bayh-Dole Act. The next one, and then we're done with legislative acts. This is actually the NIH Road Map. So Zahironi, I'll give you a little offline background. The University of Rochester discovered a new target, a new gene, a new protein, cyclooxygenase 2, COX-2. So COX-1 is what aspirin works on. It blocks the production of prostaglandins, and it's a great painkiller and anti-inflammatory particularly, although I'm not 100% sure they understand exactly how aspirin still is a painkiller, but certainly anti-inflammatory. So University of Rochester saw that in inflamed conditions, and even under proliferative drive in cancer, there's another enzyme that becomes expressed, induced, upregulated, COX-2, and they said, if we got drugs that were selected for COX-2 and not COX-1, they wouldn't cause ulcers, they wouldn't cause blood thinning, which can be a safety problem, they would be great anti-inflammatory and analgesic drugs, and they patented this in a very strong, very complete patent. Well, Merck read the patent, and Searle, and it's just interesting stories, I don't have time to, and because I was at Searle at the time, and they said, wow, that's a great patent. Let's go develop that assay, and let's find a drug, and let's develop COX-2 inhibitors, which Merck and Searle did, and I'll have a little overhead on this, and then Rochester kind of waited, took a while for their patent to issue, but then in 2002, May 1st of 2002, they finally got their patent, and on May 2nd of 2002, they sued, they didn't want to go after Merck at the time. They thought Merck was too big, so they went after Searle, but then Searle got bought by Pfizer, so they were dealing with the biggest pharmaceutical company on the planet. The end story is that a judge invalidated Searle's patent. And the reason why is he said that Searle just taught how to find COX-2 inhibitors like Celebrex and Vioxx, but they themselves never did. They never reduced it to practice, and they never set up a screen. And they said if you only would have found one molecule, if you would have tried to screen and showed that you tried to enable your technology, it would have been a different story. So this concept, we have all these targets and we don't reduce them to practice. We need to screen to find a prototype molecule that enables and validates the technology, is one reason that it led to the roadmap. And this is a number of cells that deal with, among bioinformatics and computational biology, which is critically important, also building blocks and molecular libraries, and this actually deals with high throughput screening as well as public-private partnerships. So Zahironi was pretty enlightened, and nobody knows how he came up with this. He became director of the NIH, I think, in like the spring of 2002, and in the fall of 2003, he sprung this, and it seems he spoke to maybe 200 or 300 leaders in academia, in the clinic, in science, technology, and try to understand what should he focus on, what should the NIH focus on, and I think what he learned is this University of Rochester lawsuit was making academics very nervous, the tech transfer offices, and also there was a fear that pharma was failing to deliver not only medicines in general, which was only partially true at the time, but especially for neglected diseases, non-commercial diseases, malaria, tuberculosis, even amyotrophic lateral sclerosis, where there wasn't a big market. So he came up with this re-engineering kind of translational research, not just basic, but to translate the basic research into improving human health, and of course pharmaceuticals is a fantastic way to do that. And it still continues to evolve in 2012, despite the budgetary pinches, constraints that I showed you. I guess I showed you that slide earlier, so you're seeing this is not my final overhead version. So it's a constant craving. There's the statistic I was looking for, 30% drop in R01 awards from 7,500 a year in 2003 to 4,900 in 2013. So historical, I don't know if you could see this back there. I can see it very clearly here. I'll just put some highlights. You know, the pharmaceutical industry started on a horse and buggy, drawing a wagon, selling what are called snake oils. I mean, they were literally oils taken from snakes that folks were saying, sellers were saying, will cure all your ills. I think World War II is the transformative year. This says pharma companies apply chemical libraries in drug development. So this, what we thought at the time in the 1990s and 2000 was something new and great, was actually around right around World War II. And why? It's because after the discovery of penicillin, people got the idea there must be other types of biotics in soil, in plant tissue, in the ocean that could be transformative as well. So Japanese companies in particular industrialized this. They collected and fermented samples from all over the world, and there were dozens and dozens and dozens of antibiotics, certainly. I don't know about more than 2 dozen or 3 dozen, but probably about 2 to 3 dozen antibiotics approved out there. So this was a turning point. At the same time, a little bit after that, the thalidomide incident in the United States. So this was antibiotics. We can discover drugs more efficiently than ever before, and then regulatory pressure saying, now hold on a second, you know, first do no harm. And the fact that thalidomide wasn't approved in the United States and we avoided that disaster, those are the 2 milestones I think can really define the pharmaceutical industry today. The first biotech companies, you know, 1980, Bayh-Dole Act didn't take scientist entrepreneurs long to take advantage, in a good way, of the Bayh-Dole Act. And I think another transformative, and this is off the, just an arrow pointing, that shift to new targets and engineered proteins, focus on novel drug discovery technology, and around the early 1990s instead of classical pharmacology. So we'll go further and talk about how a drug gets to be discovered. This is kind of going backwards. So pharmaceutical companies are really good at clinical development. That's what they do. They submit an investigational drug, you know, phase 1, phase 2. Phase 1 is safety testing. First, do no harm. Phase 2 is a small trial. You don't want to spend a whole lot of money on a large trial if you're not sure it's efficacious. But once phase 2 tells you that there's efficacy, you go to a larger phase 3 trial most of the time, not necessarily always. Phase 2 and 3 can be combined sometimes. And actually phase 1 and 2 can kind of be combined, depends on what the disease is and what the drug is you're trying to go after. You submit a new drug application, and then you get FDA approval. This process, Searle, so G.T. Searle and Merck, the paper on COX-2 came out in 1992 and 1998. Searle and Merck both had a COX-2 inhibitor that was about 6 to 7 years. No one gives this the cred that it's due. That like broke records to go to a first-in-class medicine for a chronic indication where safety is a big deal in under 7 years is absolutely remarkable. It normally takes 10 to 14 years to pull that off. So this process is somewhere on the order of 7 to 10 years. and it's like a battleship. Once a pharma starts on a development path, they don't want to turn back. And the more money they invest, the less willing they actually are to say they made a mistake, which might explain the high failure rate I'll show you soon. Drug gets discovered. I think many of you know there's some pathological basis for the disease mechanism. You express the reagents, the targets, you engineer them into a cell or just engineer them and overexpress them. You can do biochemical or cell-based testing. You'll get the hits, the actives, the inhibitors or the activators. You'll usually refine them somehow for potency and selectivity and safety. You'll put them into an animal, and you'll then submit that under regulatory supervision for testing in humans. And this part here is certainly, all this can be done by universities, but usually universities stop as soon as it gets to like a formulation part to make the drug. What is formulation? Make it soluble, make it bioavailable, have the pharmacokinetics being right. And this is usually should be very flexible. New targets are always being discovered, new assay technologies are always being discovered. So the folks in this room are really smart, dedicated, innovative, and flexible, and the folks in pharma are business-oriented. They want a return on investment, and they don't want to make mistakes. And more importantly, I don't think they want to admit they made mistakes either, because it's a business. I'll send you this overhead later, Janusz. They're too small type here. I'm seeing now. But there's some great language here. If any of you ever have ideas about going after a new target, there's now special categories of grants that encourage this. And I just came across this overhead somewhere on the web. It just had great language to put into the pitfall section of a grant. But we'll move on because I just explained the stages to you earlier. Now we're going to get to pharmaceutical R&D. So, it's really takes wheel buckets full of money. And it's almost like gambling. It's worse. Well, I don't know if it's worse. You'll see. It's like gambling. These are overheads. You see these motivational overheads that say all sorts of good things. I kind of like the ones that say kind of not so good things. So, futility. You always miss 100% of the shots you don't take, and statistically speaking, 99% of the shots you do. That's futility. When you have less than a 1% chance of success. And that's the chance of success of discovering a new drug. Any target you go after, any program, your chances are under 1% that you'll succeed. Despite the fact that the pharmaceutical industry spends more money on research by far than any other industry, I think this is still true today. It's somewhere on the order of, it's dropped from in the low 20s to the high teens. About 17% of revenue is spent on research. Just to give you an example, aerospace spends 4% of revenue on research. So tremendous amount of money spent by pharma. Time is of the essence. This is showing two drugs here, Adderall and Tagamet. These were created by the father of medicinal chemistry, the father of the modern pharmaceutical industry, Sir James Black, who had the idea of rational drug design, to know your target extremely well and tune the drugs you're making to hit the target better and better. From Inderol and Tagamet, and these are beta blockers still used today, and Tagamet not so much, because now Nexium it has taken over. It took 13 years to come up with the first Me Too drug. Well, it's telling me 10 years. Okay, it took 10 years to come up with the first Me Too drug. And this is the time from the transformative, the first in class, to the Me Too, 6, 5, 4, 2. To the time that Searle came out with Celebrex and Merck came out with Vioxx, there was a 3-month difference between the industries. So you're spending all this money on research, and first to market usually means in any industry, it could be potato chips. If you're the first one to invent the potato chip, you have 100% of the market. The second one that comes along was only going to get 40% at best, unless you really blow it. So the brand name, the one being first, represents billions of dollars of revenue to a farmer just for being first, even if there's only a 3-month gap. So we come to today. What's the status of pharma today? There's something called the patent cliff, and what I'm showing here is spending in pharma going from 95 to about 0607. This graph was handy, constant upward, but these are the number of drug approvals. Appears, peaking at 97, 98, and then going downward to about 0.6, and that hasn't changed. It's about 22, 23, 24 drugs approved a year. Here is the attrition rates going just for industry, just for the clinic, where from phase 1 to phase 2 to phase 3, you're talking about somewhere on the order of at best a 10% success rate in the clinic. So 1 out of 10 succeed once you move them into the clinic, and actually only 1 out of 100 or less succeed preclinically. So you can do the math. Because of this, I think if you don't manage your portfolio well, it takes a long time to develop drugs, and if you pick the wrong targets, or they're very difficult targets. You're trying to fix cardiovascular disease beyond channel blockers and beyond statins, and you don't succeed at doing that, you end up losing patent protection for your best drugs and losing tremendous amount of revenues. And this is billions of dollars here going, this is 40 billion on the top. So you were talking about 100 billion loss in revenue just the period of time of 2010 through today. That's actually about 15% of worldwide sales of pharmaceuticals. So that's a tremendous loss to any industry to lose in a period of 4 or 5 years 15% of your revenues, and altogether it may be as much as 20% of the revenue. So there's been a tremendous downsizing in pharma. I probably don't have the time to go through this in detail, but you heard of the saying lies, damn lies, and statistics. If you actually look at from 1963 to today, 2008 roughly, and you take a best line, of course there's no correlation here, this is a flat liner, and that's the point. The number of drug approvals, effectiveness, defining as getting the job done, has been constant in the pharmaceutical industry for decades. Efficiency is defined as getting the job done at a reasonable cost per unit. That's where there's a market decline, because you can see the tremendous increase in budgets starting about in the mid-1980s. But for a while it looked like there might be an increase in innovation of the number of drug approvals, but we see now that that had zero impact, despite going from about 4 billion to about 60 billion, despite a 15-fold increase in pharmaceutical R&D spending over this period of time. There has been no change in the number of new drug approvals, looked at over time, and if anything, the more money that's spent, it seems, the worse the situation is. And it just so happens that if you start to see a bad situation in 1998 starting, you have to look back 10 years, because it takes 10 years at least to develop a drug. And it's right in this time frame that high throughput technologies, the radioligand binding assay was developed, so they started to use radio-labeled drugs to do binding assays and enzyme assays. That's the time when pharma moved away from whole animal assays and into the microwell or into the test tube. And many think that's a factor, but top dozen blockbusters, it's interesting that only 6 of them are small molecules, right? The other 6 are biologics. So there are just all these statistics supporting that there's an innovation gap in pharma. Interestingly, 3 of those small molecules are CNS drugs, and almost every pharma has shut down CNS because it's very expensive. They're usually chronic indications. They don't understand behavior of the brain well. So they've shut down their central nervous system diseases research department, and yet, if you look at the blockbusters, half of what they have is central nervous system. I don't understand the paradoxes, frankly. And then there's one each, only one for gastrointestinal, cardiovascular, and asthma. And this year, only 4 small molecules are in the top 10. So the situation is not getting any better. You can't see this, but one reason the situation isn't getting better is the Me 2222 days are over. This is a list of 40 benzodiazepines. So at one point, as long as you showed your drug was effective against what? Against the placebo, you could get an approval. The last 10 years, the FDA is saying, no, a placebo is not good enough. If I already have 5 drugs in this category and you're the 6th one, I want to see that your drug is better than the class leader. So pharmas are not going past 3 or 4 anymore. 40 is a little excessive. It means you're going to be selling based on marketing and not based on science or real healthcare impact. One overlooked cause is mergers. just showing we'll have to really focus on the details here, but at around the same time that high throughput technologies were coming to the fore, this is about 5 to 10 years after the Bayh-Dole Act, companies started to buy each other out, thinking bigger is better. And I think we know that that's not really true for innovation, and it still goes on to this day. It's almost like a jungle story of one pharma consuming another pharma, and then of course letting go of about 20% of the people. And you go through several rounds of this over a period of 10 years, and there's an awful lot of veterans out there who were downsized, decided to take early retirement, and have joined academia and are setting up drug discovery centers at major institutions. So public-private partnering is a solution to contain cost and enhance innovation. And one of the key questions still is, we have all these millions of chemicals, any one of them can be the blockbuster drug of the future. What's the best way to find them? So I had the rationale I presented to Pharma that the best way to find them is not to let them sit in your repositories. That's like a plane on the ground, just a depreciating asset, but to move them through assays throughout academic laboratories. So the rationale was that academia is great at research. They don't have access to these so-called crown jewels chemical libraries that pharmaceutical companies have compiled for the last 100 years. There are so many targets, it's almost like noise, and that unless you have a proof of concept compound, pharmaceutical doesn't pay attention. And to get that, you need medicinal chemistry, and universities are weak at that. The very things that academia is strong in, pharma is weak in, plus getting return from their assets, from return on investment. I'll show you that the more pharma screens, the more money they burn through. So why not take those assets, give them to universities who desperately want them, and let them discover drug leads, and then you can go after the targets that seem to have the best traction. And in fact, high throughput screening papers, if you look at the top 10 publishing groups, where they're from, in the beginning, only 10% of them were from universities, and by the beginning, 1995, 1996, and now almost 70% of the labs publishing the most papers in drug discovery are coming from academia, not industry. That's a pretty interesting transformation in a period of about a dozen years. I don't know. How am I doing on time? How much time do I have left?
Almost, well, it's noon, 11:55.
And what time should I be finished?
So I still have some time. Hold on.
I have about 20 minutes left.
Yeah.
Let's see if we can wrap up, kick it into even a higher throughput mode. This is an interesting overhead. They crowdsourced. So the Road Map was going on for 5 or 6 years. They published an awful lot of small molecule drug leads in various papers. So someone had I think it was Nature Reviews in Drug Discovery, there is Nature Chemical Biology, took 12 experts from industry and academia who were great medicinal chemists, well known, and 2 of them are my friends, and said, look over these publications and tell us what you think. And it's just interesting how chemists, you think, would be able to agree, yeah, that's a great lead, that's developable. Red means it's not a good lead, stay away from this. Blue means, yeah, that's pretty good. So, and if you look across, if there's the opinions of the folks, of the scientists, and what's missing on the axis here is a particular chemical structure. And I think there were about 50 chemical structures that were reviewed. You can see that there's a general consensus that, not tremendous, but it's mostly red here, that most of the stuff being published is junk from universities because they're acquiring libraries, they're not the drug-like libraries pharmaceutical companies have. And then they start to agree more up over here where it turns mostly blue, but kind of in the middle it's a matrix. So to me it's a little frightening that 10 people considered the leaders in pharmaceutical R&D, medicinal chemistry, have such great debate amongst themselves on what is a good drug lead and what isn't. Just testifying, it's an extremely complex thing, a billion dollar molecule. And so my hypothesis is, what if universities had access to the very best pharmaceutical company libraries? You ask each pharma, you have 1 million compounds, you can pick 1 out of 10 or 1 out of 20 and create a library and pass that to academics. They won't know what the structures are, it'll be blinded. If they find an interesting hit and an interesting target, they'll come to you, and if you think that looks good, you'll sign a licensing agreement with the institution. The institutions were willing to do this without getting paid up front. They just wanted reimbursement for their out-of-pocket costs, so they were willing to take a chance. And that's what my hypothesis was, that if pharma does this for a period of 5 years, they'll get tremendous return for their investment. I came up with this poker logic for pharma, the 3 golden rules. The first one is know thy target well, and I think it's really academics that discover and prosecute and research all the targets. So you really want to work with the academics. Know when to hold, resource programs effectively, and know when to fold. This is the problem in pharma. For decades, people have been talking. They can't fail cheaply and fast. It's not a matter of success, because we see that it's constant. Since 1965, the number of new drug approvals have remained the same. It's how much you spend to get each one that's the problem. So only if you could fail faster and cheaper, you could have a broad impact across the pharmaceutical industry, and one way to do that is outsource. But don't write a check for the outsourcing. It's the academic institutions that could really use more revenue now. Let them do this for you. So we started a collaboration, tried to get many farmers involved. Only one said yes. We won't say who that one was here. And we went through this model that I just explained to you. The universities screen at no upfront cost except out of pocket. And if interesting discoveries are made, pharma can license them. One pharma gave us a beautiful library that we worked on, conceptually for about 4 or 5 months, that had every important element in their large file of 1 million represented by these 1 out of 10. I thought this would be like a tabula rasa, if that's what it is, a philosopher's stone. That pharma has their libraries highly annotated with biological data. So if you find a potent molecule and pharma looks back and says, that's interesting, that's never hit in any assay before. Out of 300 assays we run, this has been tested, it's been clean, that's pretty exciting if it's hitting potently on your target. So I thought the pharmas have the molecules, they have the biodata. This would just be perfect to run through dozens of assays. I set up a system where some pretty sophisticated screening infrastructure was put into practice, including this way to screen libraries in a highly efficient pooled format. Instead of screening, if 99% of the compounds are inactive in an assay, why screen every one? Why not pool 10 compounds in a well? In World War II, they came up with this concept. You had to be free of infections, particularly syphilis back then, and they knew that the syphilis infection rate was less than 1% in the population, so before they drafted you, they take a blood sample. They could combine 60 blood samples into one and test them. And if they all tested negative, all 60 of those men were inducted in the Army. If that tested positive, they would then split pool. They take the 60, they combine it into 2 sets. They take the 30 and pool it, and then 15, and so on and so forth until they found the infected person. And as they passed pools, all those people would be drafted. So I applied the same thing for drug discovery. I just put 10 compounds in a pool. I don't have time to go through this interesting orthogonal approach where I don't have to do split pooling. Since I pool in 2 different directions, every compound is represented twice, but in each pool there are 9 different compounds. So if a test tube sharing only 1 compound, 2 of them are active, I know that compound is the one. So this is all a thing of beauty. 20 universities, including University of Nebraska through Janusz, agreed to join this consortium. And what was the outcome? It's total failure. I really tried hard. Everything was working fantastic, but I couldn't get any of the pharmas to come on board, and eventually the pharma that said yes backed out after 2 years. They had a portfolio of almost 40 targets that were attractive. They actually approved some of them, and then they had to cancel the whole thing because what were the reasons? So why did this fail? The downsizing, disruption, and distraction in pharmas. They really couldn't pay attention to external innovation when they were downsizing their own workforce. That crown jewel angst. They didn't want to let anyone else wear their crown jewels even for an hour. They were worried about loss of control. These are all things I heard from the different pharmas. They're scared of you guys here. They're scared of intellectual property encumbrance. They're worried. You think you found something. You're very excited, but they realize, This is one reason they don't give royalties for early stage discoveries, because they say, if I promise the institution a royalty and then I back out of that program for whatever reason, it's a lawsuit. The institution says, you have to compensate me now because we were expecting all these royalties in an industry that has a, less than a 1% success rate. So papers have come out recently from Pharma, Bayer, and I think Amgen, that they say that 50% of the targets and the data on targets, not all data, but related to drug discovery, they can't seem to reproduce in their own shop. I say it suggests maybe they're not very good at setting up target assays, and also that the papers don't always describe the minutiae. It's like, looking at a recipe and saying, oh, I followed the recipe, but it turned out lousy. You're a lousy cook, you didn't write a good recipe. Nobody would come to that conclusion. They'd say, no, you have to have some intrinsic knowledge to cook something well, even though the recipe is still spelled out. That's my belief why it was hard to replicate data. And they had a desire for perfect targets, tens. I don't know if it was like a study section. Everybody there, if there's one little wart in the grant these days, you don't get your grant. The cutoff is, a score of 18, and if you get a 19, you don't get your grant. And you look at why did you get a 19? It was some trivial flaw one reviewer happened to see, and that's why you didn't get your funding. Scientists are that way. They love to be critical. So they wanted perfect targets. There's no such thing as a perfect target. So I've reincarnated this model, and this is my last few overheads, if I remember correctly, into something different. I think it's potentially exciting. And we have to first ask, don't even bother trying to solve a problem that you don't understand, because it probably won't lead to a solution. I thought the problem was you're spending too much money on the early stages of discovery where the risk is the greatest. Let universities explore, but it's not really the problem, I don't think. Some people say the problem is low-lying fruit is picked. Yeah, I can cure an infection, that's easier. I could maybe take care of gastric acidity, that's pretty easy. But Alzheimer's disease, memory loss, atherosclerosis, these are complicated multidimensional problems. And I can't do Me Too drugs anymore. And also the FDA is asking for outcomes benefit. And I don't understand the biology. So all the easy targets are done and the easy regulatory hurdles are over. That's what people say is the reason for pharma's problems. And I think it is from a hard revenue perspective. It's no longer like selling soap. You can't get away with it anymore. But from an innovation perspective, I don't think it's that. I don't think all the drugs for depression, the tricyclic antidepressants, Prozac, the selective serotonergic inhibitors, fantastic ion channel blockers, I don't think those were really low-lying fruit at all. So I don't think that argument is good. Premature resource commitment, know when to hold, when to fold. I think that doesn't deal with the science. I'll show you in a couple of slides. I think the reasons really are it's a complex disease biology that's not well understood for the diseases that don't have a cure yet, especially also the preclinical. And because they're complex diseases, you can't model them in a rat or a mouse. And also, single drugs rarely manage a disease well chronically because you get either drug desensitization or it's a multi-component type of disease. Certainly it's true for infection and cancer. So nobody tries to treat cancer with one drug anymore. So here's the reason, I think. This is, to me, was a very shocking set of data when I first saw it. If you look at, it looks like I can't see the tops too well here. I'm sorry, it's in white. What this is the probability of success. Target, running a screen, getting a hit, it's pretty good, 80%, and so on and so. Hit to lead, taking all the hits you get and picking the ones, despite that chemists might differ in what's a developable hit in an assay versus not, that's pretty good too. And then a lead to candidate to get the chemists to make any kind of molecule better, more selective. They do that pretty well too. So all this is good, but 20% failure, 25% failure, 15% failure, multiply all that. Candidate to first in humans so that you're getting ready to go into the clinic. Phase 1, it starts to become more of a crapshoot, 50-50 chance. But look at phase 2, efficacy. There's only a 1 in 5 chance that a drug will pass phase 2. So what does that mean? It means the preclinical models of disease, the animal models, are simply not valid, because something that works well in a rodent has an only 20% chance of working well in the clinic. So I think that's the problem. I mean, this data is telling you, look at where our biggest problem is. It's efficacy. It means our models aren't good. And look at the money, by the way. You think the most expensive part of this is money spent overall. You think the most expensive part were phase 3 clinical trials, and it is. $250 million in total, and $150 million for each one. And this is, assuming only 2 of them are being prosecuted. But chemistry, medicinal chemistry, because the success rates are so poor, you have to have 50 projects in the hopper in order to get one success out. All those together cost $500 million, almost double the amount of the total amount of money spent on phase 3. So these are the 2 problems. I can't afford preclinical research. It's too expensive to optimize molecules for potency and safety. And I have no model with which to do it. It's like my compass, trying to steer a ship on the ocean, and my compass is spinning around and it's midnight. How am I going to get to my destination? So really the only answer is, one would think, repositioning. Maybe look a little more carefully. Every drug has a multiple of actions. It has one dominant action, but many others. Maybe we should look more carefully. And you've heard, raise of hands, how many people have heard of drug repurposing? Just trying to get a sense. Well, I hope this talk is not too complex for everyone. It's kind of a mantra now. So instead of spending $1.5 billion to develop a new drug, let's avoid the whole preclinical research process, and let's avoid phase 1. Let's go right to phase 2 efficacy with a past drug. And my hypothesis is that the concept of drug synergy has not been explored at all preclinically. All screening does is try to find single lead molecules active against assays. To my knowledge, there's no literature on running drug screens, almost none for synergy. So all drugs have a dominant mechanism of action, but FDA-approved drugs are believed to have up to 5 off-target actions, and the contribution to efficacy is not always clear. So we've developed some computational methods that look into pooled FDA drugs and try to detect sort of binary pairs that are working in an assay only together and not alone. That's what I'm trying to get support for from not pharmas, but from some other types of companies. And the rationale for the model is that there are 1,500 approved FDA drugs. I think if you went through the patent literature, you'd find every class of drug is patented just about for every disease already. But 1,500 drugs as binary pairs, almost 1.2 million different binary pairs. So it's impossible that all the applications have been discovered or even thought of for every pair possible. So we're in the process of developing a highly efficient assay approach that's 30 times more effective than testing each pair individually. And we hope if we find combination activity in prior FDA-approved drugs that they can be promoted directly into the clinic, directly into human clinical trials. And polypharmacology drug screening in general is not relatively immature. It's like a completely immature field compared to classical high-throughput screening. We have 20 universities on board who I think would be willing to pilot this with us, including hopefully University of Nebraska. And I know Janusz has an extremely interesting assay technology. And what are the odds that a complicated assay system involving a large protein and a lipid interface, for example, that a small molecule can disrupt that kind of interaction? But maybe 2 molecules, right? One comes in and induces a conformational change and opens up an opportunity for the second molecule. It's kind of speculation. And nutraceuticals, this is just one example of berberine. Have you heard of berberine before? Has anyone heard of it? I'm looking at things like berberine and taurine and, coenzyme Q. There's some nutraceuticals that have fantastic clinical data. I don't understand it, but there's so much noise in the field that nobody takes nutraceuticals. It's like a big GNC shop where 1 out of 100 things are actually effective and have more clinical data than some drugs. So what if one combined, nutraceuticals with drugs in a certain kind of way to find things that are known to be safe, effective, and that are extremely cheap, and that there are combinations that work in a way that single molecules never could work. So in the next 3 months, I hope maybe to go to the collaborators and show them some interesting libraries and interesting rationale for it. I don't have time to get into what this power purposing, forgive these slogans up here without any data or mechanism to support them. But I guess it involves if 99% of the compounds, are inert in one screen and pharma is sitting on a lot of inert compounds, they should just donate those, don't you think, to universities and say, take this. It's like looking for oil in Oklahoma. We can't afford to do all the drilling, but if you're willing to do the drilling, here's our rig and do it for us. We think there's no more oil there. If you find it, you're doing us a favor. So the pharmaceutical industry has the poorest efficiency, I think, of any industry. Nobody could tolerate, I'm going to spend $1.5 billion overall on a project because 9 out of 10 fail, and my chances of success from the concept to the product is less than 1 in 1,000, to be generous. No one else could do that. Spend the most on R&D and be so poorly efficient, but it's that billion-dollar molecule that comes out at the end. So it's that one success can pay for all the failures. I think I've showed you just some logical data that the bottleneck is phase 2. It's efficacy. That's the problem. A dominant factor by definition, this is almost propositional logic, it has to be, it's a lack of translation between the animal models, because no one in their right mind puts something into the clinic that didn't work in an animal. You always cure cancer in rats. You've heard that so many times. So I say avoid the preclinical models and just find a way to go right to the clinic and look right for efficacy. And it could be that single drugs are not the way to do it. Maybe new combinations are the way. And that a greater emphasis on polypharmacological phenotypic drug discovery could be a logical solution. And to include, I'm calling them nutraceuticals. I don't know what the right word is, but this is a conference on Nutrigenomics. You probably understand it better than I do. So thank you very much. You all paid attention remarkably well for being right before lunch, and if there are any questions, I'll be happy to entertain them. Thanks.
Thanks, Mel. It was a great talk. We'll take a few questions now before lunch.
Thank you very much for your presentation.
You're welcome.
I wonder what would be the interest to have your drug synergy model, because in practice, for most of the complex disease like CNS, our patients are already polymedicated. So what would be the interest to sell one pill with 2 drugs when you have different drugs in different pills, and so that the neurologist can already decide what is fitting well with the patient?
So it's it's a very good point you make. About patient. In other words, patients being pre-medicated implies that we don't have a grasp on all the variables. But in clinical trials, you wouldn't let patients knowingly, you wouldn't enroll them in a study knowing they are on certain drugs already, and that's a variable that you don't control. So I think you would have to exclude patients, which is typically done. But maybe better way to answer your question is to say it could be the models are maybe somewhat valid, but in a uniform genetic organism like a rat. But now you go into the clinic, and not only do you have, let's say, prior drug treatments that could have affected the brain, but you have genetic variability as well, and behavioral variability and social variability. So you give one drug, and you reach a significance level of 0.07. That drug's not going to get approved. But 0.07 is pretty good, right? I mean, if you got 0.05, so the notion is if you went into the trial a priori with 2 agents, each one of them capable of a 0.07 or 0.1 even, the probability of achieving 0.05 with both of them might be greater. Of course, probability of side effects, you'd have to cover that aspect as well. So it's not a panacea, this. It's just suggesting that nobody treats with a single drug for cancer. It's not done. for world diseases, infectious diseases, nobody goes into the field with one. And if you talk to patients who have high blood pressure, it's very hard to find any of them that are taking just one. So I'm saying translate that concept in a drug discovery perspective. Well, that suggests my speaking time was up.
Other questions?
So is the program where pharma might share some of their chemical libraries with academics, is that, you think that's dead, you've abandoned it, or you think there's hope?
You know, so I'll answer the question directly and say it's dead. I give up. Failure is failure. At one point, part of success is, right, recognizing failure. The NIH had a program, I don't know how far it's getting, where they got compounds from pharma that made it through phase 2 or maybe even phase 3, but they didn't become drugs because of efficacy. Couldn't be safety, because then it's out. So they got a collection, I think, of 2 or 3 dozen of these, and then had a grant opportunity, R01 opportunity, to see if scientists had an interesting idea on how they could repurpose this small, high-value library of failed clinical candidates. I don't know where that initiative is, but I think the broad initiative, give me 100,000 of your compounds, and let's just hope for the best. They're very nervous about that. Select number of compounds, I get to see the project, I get to approve the project, I know all steps of the process. Models like that, I think there's still some promise for, possibly. Repositioning type models.
Other questions?
So I guess I would just like to argue that the rat or the mouse is not the best model, period. And pharmaceutical companies should be looking at better models for humans, such as swine, goats, sheep, dogs. There's a conference, Swine and Biomedical Research, that for many years has talked about the advantages of pigs as biomedical models. The swine genome is closer to the human genome than is the mouse or the rat. And faculty at land-grant institutions can do work on livestock much cheaper than at biomedical institutions anyway. So,
you know, I at first I was, as you started speaking, I had, you know, typical, let's say, critical response. It's too expensive and it's not genetically uniform. And then I said, well, wait a second. Maybe that's exactly what you want. Maybe you want to test your drugs for efficacy in a non-genetically uniform model to see, because that's what the clinic is like, right? That's what humans are like. So it's an interesting idea. What do farmers think of it, in your opinion? Exactly. Has it been? Can they tolerate the expense? You know, what are rats? Back when I was doing research, they were like a few dollars each. I think there are still mice are, what are they, $10 to $20 a mouse? 20.
right, but when you look at the amount of information you can get from a pig, you know, the amount of blood you can get or the size of a sample from a pig, and then the more molecular or physiological information you can get from a pig, it's just much more than you can from a mouse or a rat. I think there are examples, I'm not familiar with this, but drugs that have gone from rodent to human and then have failed in the human population with side effects.
I think it's a great idea if it's applied, I think, in the right time and place. Some reason society has accepted, you know, creating models of disease in rodents. If you see the direction I'm moving in, I don't think we've also accepted, you know, as a food source, you know, farming, certainly for thousands of years. I don't know if we're ready to accept inducing disease models in swine. And although for safety testing, you know, Pharma does look at swine for certain diseases, but this is once they're pretty sure they're about to go into the clinic. I think they'll look at dogs, certainly for safety, and even cardiovascular. And I'm trying to think, for certain skin, I'm trying to think, maybe, transplant rejection. I think they'll use swine as well in the very last stages. They certainly will. Yeah.
Any more questions?